Thank you for attending this session on Exercise is Medicine, Current Insights from C.M.Tipton's Scientific Grandchildren. In 1963, 54 years ago, I was in my junior year at Springfield College. Dr. Tipton taught exercise, and I became his assistant in the laboratory. The following year, 1964, Tipton was recruited to the University of Iowa, where he established a rigorous program in the exercise sciences. To be accepted into this program, the applicant needed a background identical to a scholar entering into medical school. The 22 students he recruited from 1966 to 1995, as presented on this slide here, established were essentially in the forefront that exercise is medicine. Today, 5 individuals from Tipton's program and their respective students will present themes concerning the reality that exercise is medicine. At this time, Dr. Tipton is getting ready for his 90th birthday. But due to illness, he could not be here in the flesh. His spirit will be here, and he has a message for you that Dr. Frank Booth will present. So, Ron, let us have the first speaker.
So I'm Ron Terjung, and it was my distinct pleasure to have been one of Tipton's trainees. Could you go to the next slide? There we go. So as you see, I was one of his trainees, and the design of this symposium is that some of our trainees, in this case, David Hood. Where's my next slide? There we go. Will be our first speaker. David has distinguished himself with an impressive research program. We're delighted to have him here. He's a professor and candidate chair, research chair at York University. He's been appreciated with a national, international reputation for his research activities. And as illustrated now on the slide, he's been an active mentor in his own right with numerous pre-docs and postdocs. The form of this introductory slide will be used with each of the speakers in their respective way with their mentors. And the individuals you see in italics represent the postdocs for each mentor. That's sort of the scheme we apply. So without taking any more of David's time, I'm pleased to have him come to the platform.
Thank you very much, Dr. Terjung and Dr. Baldwin, for organizing what a great idea this is. It's certainly original thought to set up a program or a symposium which honors Dr. Tipton, which I'm fully supportive of, and I wish he was here to join us. And let me just say that I'm supposed to talk about exercise and muscle health and mitochondria, and I'm certainly going to get on with that momentarily, but I just want to say that this kind, and this will not be a surprise to any of you here, this kind of outstanding scientific lineage that we're talking about here today in this symposium develops your research skills and your research interests. And it's just, for the students in the room, I think, it's just very important that you set yourself up in this way. I was so fortunate to come from this kind of scientific lineage that it rubs off on me, certainly big time, and then also, of course, I hope, on my own trainees. So let's see if it helped develop my research interest coming from this lineage of Tipton and Terjung. Back in 1968, from the American Journal of Physiology, Dr. Tipton and Terjung and Dr. Barnard published this review, I believe it was, on the role, the interaction of thyroidectomy and thyroid hormone and training, and they summarized the effects of thyroid hormone by themselves and then the effects of training. And then that must have rubbed off on Dr. Terjung, because 8 years later, in 1976, when he was his own investigator in Illinois, published this paper on biochemical adaptations of trained thyroidectomized rats to see the interaction between thyroid hormone and training and whether they were independent or not. And they certainly ended up being independent, and this was a pioneering paper in the field. Now, when I arrived in Dr. Terjung's lab in 1981, I knew nothing about thyroid hormone, and I really had no interest in thyroid hormone. I was studying leucine metabolism, amino acid metabolism in skeletal muscle. I certainly learned a lot about that. And then I learned a lot about mitochondria, because the main enzyme of leucine metabolism is in the mitochondria. So that set me up for my future career, which is related to mitochondria. And people were so surprised that I finished my degree that people in the lab gave me this banner with Dr. Terjung's picture on it with "I survived Dr. Terjung 1986," because they were so surprised that I actually finished the program. But the thought processes of thyroid hormone and exercise continued in the back of my mind, and even as a postdoc, I started studying and thinking about thyroid status and the expression of metabolic enzymes, mitochondrial enzymes, during chronic exercise. This is one of my postdoc publications. And then as an assistant professor at York, the continuing thoughts that I learned as a doctoral student on thyroid hormone were again prevalent in the work that we did, and I was studying it in terms of development. So it has a large impact on your career. Now, I'm supposed to talk about mitochondria, and I feel it's very exciting to be in mitochondrial research these days. When I first began, the thought process was only about these bean-shaped organelles that when they undergo biogenesis, then you have an improved muscle performance, generally speaking, for athletics in general. But now, of course, our focus is on health. Our focus is on when mitochondria go bad and there's some dysfunction and reduced biogenesis, and that's associated with aging and disease. And it's reflected, the interest in mitochondria is reflected in the publication growth on mitochondria, as you can see here from the year 2001 to the current time. The interest in mitochondria and publications is growing continuously here, as you can see, whereas these other organelles are, who cares about the nucleus anymore? The nuclear publications are going down. ER, nobody ever was interested in ER, and Golgi is reduced even further. So this is, again, an exciting field to be in, because a lot of people and a lot of people feel that way. So what do mitochondria look like in muscle? Well, the original work by George Brooks and other colleagues in that period of time in the 1960s, including Hans Hoppler and his colleagues, developed the thoughts that mitochondria really do not look like this. They look like this. And this is one image from the paper by Kayar et al., Hoppler's group, that showed mitochondria exist in a reticulum, a membrane network that extends throughout the muscle cell. And this has not died down in any way. This is a publication from just a few weeks ago by Dr. Glancy and Balaban at the NIH, to show the importance, the relevance of this network of organelle that exists within muscle. You can see these very impressive images in the heart. This is heart mitochondria. They exist as a reticulum with little junctions in between. They've colorized the images here to show this a little more clearly. And the physiological significance is described as these junctions can open and close, and conductivity of electrical, the membrane potential can exist, or it can be cut off, and if it's cut off, then the mitochondria undergo mitophagy. So this is an area that is still growing, and we're still trying to understand the role of the mitochondrial morphology in muscle health. How does this morphology arise? It arises because of the expression of proteins that allow it to fuse, allow the mitochondria to fuse together. These are OPA1 and mitofusins, or allow the mitochondria to undergo fission, that is, a dysfunctional portion of the mitochondria can be removed and undergo mitophagy, and I'll come back to that. And it's really the ratio of these regulatory proteins that determines whether a mitochondrion grows as a reticulum, determined by the abundance and overabundance of these fusion proteins, or undergoes fission and the mitochondria become more fragmented and dysfunctional, and that is an overabundance of these fission proteins. So we looked a few years ago at the role of exercise and the role of muscle disuse on the ratio of these types of proteins. You can see here's the two fusion proteins, two fission proteins. The ratio of fusion proteins is higher compared to fission proteins, and that leads to a reticular organelle looking like this. On the other hand, with muscle disuse, you have higher levels of the fission proteins and lower levels of the fusion proteins, and so the mitochondria end up looking like this in a more fragmented orientation. So this has relevance to health because these mitochondria have a poor membrane potential, they have low rates of respiration, higher ROS production, and that contributes to mitochondrially mediated apoptosis and a decrease in muscle mass. So understanding this is important. Now, we have here an organelle in a myoblast, and let me see if I can get this to play. How do I do that? How do I get the video to play? Beautiful. So if you look closely, you can see the mitochondria inside this myoblast, and you can see them moving around. And if you look up here, you can see mitochondria undergoing fission right there. This is over a course of 5 minutes in a myoblast. So the movements and the fission and fusion processes can be visualized very simply in a myoblast. It's much more complex in a myotube. But what we can do in this kind of situation is take frame after frame after frame, and you can look and see here's an intact mitochondria. And if you look a few seconds later, you can see that it's broken and it undergoes fission. Similarly, here you can see that these two mitochondria are separated, but then where is it? Over time, they become fused. And this is regulated by the cytoskeleton and the cytoskeletal microtubules in particular, and kinesin motors that move mitochondria around. So there's not much known about this. What happens to mitochondrial movements with contractile activity, with exercise? Well, I don't know that. But I do know that when we change calcium inside the cell, here's an increase in intracellular calcium, and then when you add EGTA, you reduce intracellular calcium. We see changes in the distance of mitochondria, so when you add calcium, they don't move as much. The speed of movement is reduced. When you take the calcium away, they move back. They start moving further and further within the cell, and the speed of movement increases as well. So understanding this mitochondrial dynamics will allow us to understand the regulation of organelle morphology in health and disease. But this is in a myoblast. We have to take this to a myotube, and it's much more complex because, of course, the mitochondrial density is quite a bit higher. Well, what is the exercise pathway that triggers mitochondrial biogenesis in muscle and an enhanced muscle health? What is this pathway that has economic and scientific ramifications? Well, we know a lot of the steps now, since we didn't over the 20 years ago, but we do now. We do know that there are 3 basic signals that trigger the activation of gene expression involving proteins such as SIRT1, P53. Everybody knows about PGC1. It's hard to avoid this protein. It's the most popular metabolic protein, I think, in existence now, and as a transcriptional regulator for mitochondria, leading to an increase in mitochondrial function and muscle health. So we can't avoid talking about this for a few minutes. Let's do that. PGC1 is a transcriptional coactivator. It leads to the transcription of many, many genes that find their way into mitochondria. When you overexpress PGC1, you get an increase in biogenesis, improved endurance, VO2 max, lipid metabolism. That's going from left to right. When you take away PGC1, you make the muscle very white. Mitochondrial dysfunction occurs. We have a reduced mitochondrial content and increased apoptotic susceptibility. So that's overexpression and underexpression. What happens? Is PGC1 important for exercise-induced biogenesis? Well, there's several papers out there that suggest that not really, it isn't. It's good for basal mitochondrial content and function, but when you impose exercise, other proteins take over. Here's an example. Here is respiration. This is state 4 respiration in the knockout, and this is state 3 respiration in a knockout of PGC1. You see reduced mitochondrial function here in state 4, reduced mitochondrial function in state 3. If you train those animals, you rescue the problem. It's fixed, even though you don't have PGC1. Fixed. So we have rescued the defect, the transcriptional defect that existed in the absence of PGC1. What did that? Some other protein did that. So which one is it? There are many proteins required for mitochondrial biogenesis and many redundant pathways, and I don't think we're ever going to find a protein that is unique and the only one that regulates mitochondrial biogenesis during exercise, because when it's not there, the others take over. Transcription's fantastic. Everybody loves transcription. What about mitochondrial assembly? Well, the assembly is complex because the mitochondria data from a few years ago suggests that there are 1,200 nuclear genes encoding mitochondrial proteins that find their way into this organelle. 1,200. Mitochondrial DNA only encodes for 13. So there's a pathway here involving precursor proteins being imported into the organelle through the TOM complexes and TIM complexes and ultimately getting refolded and incorporated into complexes in the mitochondria that's vital for function. Nobody had studied this until 1996 in muscle. We showed that SS and IMF mitochondria have differential rates of import. This is the imported protein here, the mature form of malate dehydrogenase. And in subsequent years, we looked at, and there's several studies referred to at the bottom, that showed that exercise enhances the import pathway and disuse reduces it. So this is the effects of chronic exercise on import. It's above 1. That means it's 70% higher with exercise. The import process is, and that's because they've got a 3-fold increase in the expression of protein import machinery components, those TOMs and TIMs that are important for the transport of proteins into mitochondria. Okay, so if you look at a graph, a hypothetical graph of the amount of precursor protein and the amount of import, what you're seeing here is that any level of precursor protein, you get a certain amount of import. If you have chronic exercise, then that import rate is enhanced at any level of precursor protein. And what we showed later was with denervation, the opposite occurs. Import is reduced by about 50% along with the expression of protein import machinery components, and so the curve shifts downward like this. So this is important, right? Because there's 1,200 proteins that have to go into the mitochondrion to make up a functional organelle. So there's a fairly good correlation between the protein import rate and mitochondrial content. And if you're down at this end of protein import, you've got a very low level of mitochondrial content. And the question we asked one time was, can exercise rescue this biogenesis defect? Well, the answer came from an unusual source. We were studying the Bax back knockout mouse. Bax and back are in the mitochondrion, and they are involved in pore opening and pro-apoptotic protein release. So nothing to do with import, nothing to do with respiration or anything like that. We were studying this animal to study apoptosis. And what we found was that the mitochondrial content in this double knockout mouse, the IMF area was reduced. The SS depth, you can see the depth of the SS layer here is much reduced compared to the wild type. And if you look at the IMF area, which is tough to see here, it's also reduced. So we asked the question, is it import that is affected in these animals? So it turned out to be true. So here is the import in the double knockout. You're looking at the level of this ornithine carbamyltransferase that's been transported into the mitochondria in the double knockout. It's reduced. Well, we then asked, can exercise, this is a running wheel, mice on a running wheel for 4 weeks, can exercise rescue it? And this was one of those great moments in Yuan Zhang's life. She was a Chinese PhD student, came over, did some great work, and showed that the import is basically totally rescued with voluntary wheel running. So instead of having reduced import defects, exercise rescued that problem and brought the import back up to normal. So this is, again, important because you've got 1,200 proteins that are traveling through this route. What about mitochondrial quality control and turnover? I picked this out of the literature. It seems like mitochondrial quality control is now being thought of as a therapeutic target. What are we talking about by mitochondrial quality control? We're talking about not only the biogenesis, but also the mitophagy of it. So we looked at this in the context of denervation. Here is the perineal nerve, which we would sever in a rat and leave it for several days for the muscle to atrophy. And this is how do I get that to activate that little film there? If you look at the muscle here, the muscle is contracting, and he's got a little backpack on his. Could you play it one more time? Thank you. This is chronic stimulation on one leg, and the other leg is the resting leg. Okay. So let's consider this turnover in the context of those two conditions here. So the mitochondrial life cycle is shown here. We have signals that lead, as I said, through biogenesis, through PGC1 to mediate biogenesis. We have a healthy mitochondria, and it's going to fuse to the reticulum. And this is our steady state content of mitochondria here, a reticulum. When a portion of that mitochondrion becomes defective, loses its membrane potential, etc., it undergoes fission. These fragmented mitochondria have high reactive oxygen species, don't produce a lot of ATP, and they need to be removed. So they are tagged by autophagy machinery proteins, which I'll just mention on the next slide, and they undergo this process of degradation in the lysosome. So this is an important organelle that I'm going to focus on in just a second. The mitochondria have to be transported there, and this is called flux, mitophagy flux, whereupon it is taken up by the lysosome and degraded. Now, in a denervation model, mitochondrial content goes down. So our hypothesis is that mitophagy, the degradation part, is greater than the biogenesis. We already know biogenesis is impaired. We know that in a denervation model, and we hypothesized that mitophagy was accelerated. So we did some experiments, very complicated slide. This is the tagging of mitochondria. Here are the proteins that are associated with mitochondria when they undergo this mitophagy. They include proteins like P62 and LC32, and you measure these on a Western blot, and when you see that association on isolated mitochondria, you know they're targeted for mitophagy. And we look at proteins like LC3 and P62, and we look at the flux under denervation conditions, and our hypothesis held up. That is, mitophagy measured either way is associated with an increase in degradation rate. The problem is you're still left with dysfunctional mitochondria in the muscle. So the remaining mitochondria are still dysfunctional. You can see that by reduced rates of respiration. This is normal here. At 3, 7, and 14 days, you can see lower state 3 respiration and higher reactive oxygen species despite an increase in degradation rate. So it's clear to me that mitophagy is accelerated, but it's not enough. It's not accelerated enough to get rid of the dysfunctional mitochondria. What's going on? Well, if you look at electron microscope pictures, I have no idea what time it is. Yes, I do. It's 20. I still got time. Good. So this is a control muscle, and you see none of this. What is this? This is lipofussin. Lipofussin is an accumulation of debris inside muscle, and you see this very abundantly in old muscle, and we see it in short-term denervated muscle. There's an accumulation of organelles, lipid droplets, and other aggregates inside, very difficult to degrade. And it's a sign of lysosomal dysfunction. It's an indicator of lysosomal dysfunction. So what we have with denervation is we have an increase in mitophagy flux, but the clearance of mitochondria is impaired, evidence shown here, leading to lipofussin accumulation. So what we end up having is reduced amounts of mitochondria in denervation, but an accumulation of dysfunctional mitochondria in which respiration is poor, ROS is high, and protein import is impaired. Well, exercise to the rescue. Here we have a model of, again, the model of chronic stimulation. I showed you the rat with the limb contracting. Lysosomal adaptations occur pretty early. as a result of chronic stimulation. We've become interested in this because we're looking at an aging model, we're looking at a denervated model, we're seeing all this lipofuscin, and that tells us that there's something wrong with the lysosome. So we decided, my postdoc and I, Yoo-Ho Kim, who's going to take a position at NIH shortly, he's been with me for 2 years from Korea, done an excellent job characterizing the lysosomal adaptations to exercise. So the main driver of lysosome synthesis is a transcription factor named TFEB, which drives the synthesis of lysosome-related genes. What are some of those? They're not, household names in exercise physiology. These are called LAMPs. These are lysosomal proteins. Cathepsins are degradation enzymes. This is a channel mucolipin for calcium extrusion. Anyway, all markers of lysosomal function and biogenesis. So we looked over the course of time, 1, 3, 7 days of chronic stimulation, and compared those adaptations to mitochondrial adaptations reflected here by cytochrome oxidase. If you look at these Western blots, there's a pattern that emerges. The control is here, there's 2 animals, and then this is the chronic contractile activity. If you look at 3 days, and you just simply look down here, 3 days post the onset of contractile activity, what you see is more intense bands pretty uniformly throughout at 3 days of chronic contractile activity for all of these important lysosomal proteins. The graphs are shown for a few of them here. So 3 and 7 days are marked increases in markers of lysosomes, suggesting, and if we do immunofluorescence on single fibers, the slide I forgot to put in here, you can see an accumulation visually when you're looking at the single fibers. In comparison, a marker of mitochondria goes up a tiny bit later. This is not quite significant. I don't know what happened to that number, but anyway, not quite significant at 3 days. 5 days and 7 days are where you see an increase in mitochondrial content. So we surmise, interpret, that the lysosomal adaptations are occurring earlier than the mitochondrial ones. So this increases lysosomal capacity and potentially can reverse lysosomal dysfunction associated with muscle disuse and aging, potentially, because again, in aging muscle, I didn't show you the picture, but in aging muscle, we also have this accumulation of lipofuscin. So, Dr. Baldwin said earlier about exercise as medicine and the theme, of course, of ACSM. I think I could be a little more specific. Exercise is mitochondrial medicine. Exercise definitely mitochondrial medicine. I've given you 3 examples in this brief talk. Exercise rescues transcriptional defects associated with PGC1 deficiency. That's certainly true. Exercise reverses protein import defects, leading to more mitochondria. Exercise increases lysosomal biogenesis and degradation capacity to help clear dysfunctional organelles. I am so grateful to have been part of this lineage, and I hope that I carry on the tradition of great mentorship with the students that I currently have. This is the Dr. Tipton's scientific great-grandchildren. I'm going to educate them in this lineage and hopefully set a good example for the group that I have now. Thank you for listening.
Thanks. Wonderful job. Can we switch to the Caiozzo presentation? Our next speaker is Vince Caiozzo from the University of California, Irvine. As you can see here, he is one of my prized individuals, and he is now a key leader in the School of Medicine's program concerning exercise physiology. Vince has a dual appointment in physiology and biophysics, as well as orthopedics. He's a busy man. Importantly, over the last 15 years, he has had a NIH rare P32 grant that is over 15 years old under his supervision. And he's a major person for not only the the School of Medicine, but for the undergraduates that are now in a new program in exercise sciences. So we're doing a good job at UCI. Vince, take it over.
Well, I'd like to begin by thanking the organizers, Dr. Terjung and Baldwin, for inviting me to participate in this event honoring Dr. Tipton. It's an incredible pleasure to be here. Before I get going into the talk, I just want to highlight a couple individuals. Over here, you can see some of the trainees that we've had in the lab, and a couple of them I want to highlight. There's Yifan Yang, who's in the back here, and I'll highlight some of Yifan's work. Also, Josh Cotter highlights some of Josh's work. And then also, I'm proud to note that Eric Rivas has just recently accepted a job at Texas Tech, so we look for great things from Eric as well. So how do we fast forward on this? There we go. All right, so yeah, we're here to honor Tipton, and I want to acknowledge his contributions to the field in space exploration and his involvement in such central agencies such as NASA and the National Space Biomedical Research Institute. And as I go through the slides, what you'll see down at the bottom, hopefully this won't distract you too much, but what I wanted to do was highlight some of Dr. Tipton's accomplishments and his role in helping guide the National Space Biology Program, which is really quite impressive. In going through my talk, there's going to be what I'll call 4 guiding themes of the presentation. The first is I like thinking about design constraints as a conceptual basis for thinking about physiology in general, the adaptation to activity and the adaptation to inactivity. And of course, in thinking about microgravity, we're thinking about the unloading that's induced as a result of exposure to microgravity. The second theme is that I want to propose, or maybe just kind of add on to what other people have kind of built previously, and that is not to think of skeletal muscle consisting of many separate compartments. And I'm going to try to make the argument that, in fact, the mitochondria, the sarcoplasmic reticulum, and the sarcomere are actually all linked together as a single unit, which I'm going to refer to as the SMART unit. You could come up with whatever acronym you want out of sarcomere, mitochondria, and sarcoplasmic reticulum. But the point that I want to get at, and that I'll try to emphasize, is that when we look at processes like atrophy, there's a proportional loss of the mitochondria, the sarcoplasmic reticulum, and the sarcomere. They're not disproportional. They are lost in equal proportions to one another, and that raises the interesting issue of whether, in fact, they are a single unit unto themselves. A third issue will be what's the ability of exercise to mitigate the deconditioning effects of microgravity. And then finally, of course, we want to pay tribute to Dr. Tipton and this concept of the right stuff. So in our undergraduate class in exercise physiology, we talk about design constraints all the time. And one of the models that we use to emphasize this to our students is one developed by Weibel and Taylor, where they looked at the oxygen transport system and the ability to deliver oxygen to skeletal muscle. And basically it consists of 5 steps. The first is gas exchange. The second is gas exchange at the pulmonary level. The third, gas transport. Fourth, gas exchange at the tissue at the periphery level. And then finally, cellular respiration at the skeletal muscle level. And on the basis of my talk, what I'm going to do is I'm going to focus really on skeletal muscle and think about design constraints as they apply to skeletal muscle. And I would argue that the ultimate design constraint is the force-velocity relationship. People have known about the force-velocity relationship for well over a century. But basically, the nice thing about the force-velocity relationship is it basically dictates for us what I'll call the theoretical boundary of muscle performance. Muscles can operate on the force-velocity relationship during single maximal contractions, and then as cyclic work is imposed on the muscle, it's forced to move further and further from this. If we sit there and think about the design constraints of the force-velocity relationship, we know that it's the number of sarcomeres in parallel that ultimately dictates maximal isometric force, and it's the type of myosin isoforms that dictates maximal shortening velocity. This force-velocity relationship is also important because it defines or it dictates the power that a muscle can produce. By extension, it dictates the amount of work that a muscle can produce. It also dictates the efficiency of the muscle. And really, what's quite amazing is to sit there and think, if you can play this movie for me, that ultimately at the end of the story, it's this molecular motor that imposes the demand on the cardiovascular system and on the respiratory system. And if you sit there and think about it, we've got the Tour de France coming up, and if you sit there and think about what these world-class cyclists are doing, they're literally burning up about 10 to the 20th molecules of ATP per second. In other words, there are 10 to the 20th of these events occurring per second for 5 hours a day, which is really quite remarkable. Now, of course, as I mentioned, as muscle is asked or required to perform repetitive contractions, we can't be thinking about the molecular motor itself. We have to be thinking about other aspects that become important design constraints. So here's our force generator, the sarcomere. We could think about it at a more elementary level and think about the individual myosin molecule. We've got the on-off switch, which is the sarcoplasmic reticulum. And then, of course, we all know that it's the mitochondria that's responsible for providing the source of ATP for the calcium pump of the SR and for the myosin molecule itself. The question is, what happens to each of these components when muscle is unloaded? And sad to say that while we know a lot about what's going on with the sarcomere and the force generators, we know much less about what's happening to these components. Here's some data that was generated by Dr. Greg Adams. This was published in JAP back in 2003. Really a nice data set because what Greg did is he actually went through the literature and looked for various models of unloading. And so he included data from flight experiments, bed rest, immobilization, and unilateral lower limb suspension. And then he plotted the data. This is data looking at the loss in maximal voluntary contraction strength versus the duration of unloading going out to about 180 days. And you can see that initially there's very rapid loss, and then it seems as though maybe the loss in muscle strength plateaus at right around maybe about 100 days or so. In taking a look at changes in muscle size, we're interested in muscle size because that's reflective of the sarcomeres, which make up about 80–85% of the total cell volume. Here the data is not nearly as clean, and it only extends out to about 100 days. But we can see that there's a reduction that ultimately reaches about 15% or so. We really don't know what happens beyond that. And of course, when we sit there and think about a Mars mission class type of flight, we're talking about not going out to just 100 or 180 days. We're talking about going out to 1,000 days. So that's completely unknown territory and is a huge question mark. Now, this figure is supposed to represent a muscle fiber in 3 different physiological states. The normal state, which I've shown here, a hypertrophied state to your left, and then an atrophied state to your right. And we know that with atrophy, there's a loss in the absolute amount of muscle mass. But the question I would pose to you is what happens to the relative proportions of the myofibrillar volume density, the mitochondrial volume density, and the SR volume density. And the data that I'll show you tends to suggest that there's an absolute loss in the amount of each of these components, but the relative proportions stay the same. And this comes back to the idea, provides evidence that in fact, These are being assembled and disassembled in equal proportions, and the reason for that is you can't have a sarcomere without a mitochondria and SR. Why would you have mitochondria without a sarcomere? Why would you have a sarcoplasmic reticulum without a sarcomere? There are known tethered proteins, for instance, the mitofusins, that are known to tether the mitochondria to the sarcoplasmic reticulum. The lateral sacs of the SR are tethered to the sarcomere at the M line through proteins like ankyrin 1.5. So here's some data showing what's going on with the mitochondrial volume density, myofibrillar volume density, and sarcoplasmic reticulum volume density under a variety of different physiological perturbations, and you'll notice that there's absolutely no data for the SR. So that's a big black box. If we sit there and take a look, this is a study by Ferretti et al. That was published in J Physiology after 37 days of bed rest, and you can see that there's a decrease in the mitochondrial volume density, but I would actually argue that when you collectively take a look at the overall data, it's hard to really come away emphatically saying that there are huge changes in what's going on. You might sit there and say, well, relatively speaking, there might be a 10%, 20% change, but I'm not sure that our techniques have the fidelity to really make those types of arguments. If you sit there and take a look at the myofibrillar volume density, doesn't change with atrophy. If we take a look at exposure to hypoxia under different conditions, no change in mitochondrial volume density. Again, no change here, no change in the myofibrillar volume density. Here's an aging study. No change in mitochondrial volume density, no change in myofibrillar volume density. So the argument is that we're losing these components in an absolute sense, but on a relative basis, their changes are always proportional to one another. And I think the reason for that is that it's possible that, in fact, there's a single unit that I will refer to as a smart unit. Now, coming back to our force generator, we actually have a fair amount of information on this. This is data generated by Dr. Baldwin, and this is 7 days of unloading and the change versus control, and you can see that there can be losses in muscle mass that are very rapid. Now, how does this correspond to things at a more mechanistic level? Well, to take a look at this, Ken and his group looked at what was taking place with the actin pre-mRNA and also the slow type 1 myosin heavy chain pre-mRNA. And you can see that after just basically 1 to 2 days of unloading, there's a 60 to 70% reduction in the pre-mRNA transcript. Many of you have seen figures like this in a multitude of publications. We've got our anabolic side of the equation over here acting through things like IGF-1, AKT, mTOR, P70S6 kinase. And then we have the degradation side over here involving things like atrogen and FOXO. But one of the points I want to try to make today is that we know a lot more, I think, about this side of the equation than we do on this side of the equation. Specifically in reference, if you buy into this idea that there might be a smart unit and that the mitochondria and the SR and sarcomeres are built and removed as a single unit, then what I would argue is we need to know a lot more about all the factors over on the degradation side and how they impact these ultrastructural proteins that link the mitochondria with the SR and with the sarcomere itself. Moving forward and talking about exercise, so at UC Irvine, we've been very fortunate to test a number of interesting devices in this regard. Dr. Yifan Yang, who's in the back here, worked on the space cycle, and she did a lot of nice work. If you could go ahead and play the first movie for me. So here is our first version of the space cycle. This is a short-arm human-powered centrifuge with the goal of being able to generate artificial gravity. And believe it or not, we did have a couple crazy subjects that got up to about 7 Gs, which is really quite amazing. We then came along, and through Ethan's efforts, we modified our space cycle so that not only it could have an aerobic component to it, but also you could use or perform resistance training simply by using artificial gravity as the loading mechanism. So if you go ahead and play that for me.
We're recording.
Here I am on this, going at about 3 Gs to 4 Gs, and really quite impressive. And the amount of loading you can get on, I was up to 5 Gs on it, which is really quite spectacular to feel that. And I'm not sure what Ethan ever got up to, but she could vouch for what I'm talking about. She might say that maybe we could also rename it the Vomit Comet. She did have a couple episodes like that, but she was a really good sport about working with the subjects. And then if you could show this third video, this is Josh Cotter, who's here in the front, working on basically a modification of Dr. Tesh's yo-yo device. And this was modified so that not only it could be used to subject the individual to resistance training, but also to provide an aerobic component. And ultimately, as we move forward, it's going to be devices like this that have a relatively low volume requirement, I think, that will make the mission out to Mars. I just don't see things like the space cycle fulfilling the requirement. So let me move forward here. Here's some data from training on the space cycle, doing the hypergravity resistance training. So we had a group that did squat training in a normal 1G environment using external weights. And following the training, there was a nice effect here in terms of strength gains. And then using a hypergravity training paradigm that provided the same loading on the muscles, we got virtually the same type of response in terms of the training effect. If we take a look at molecular markers, looking at things like IGF-1, here's the the group that trained with the free weights. Prior to training, there's a nice robust increase in IGF-1 post-training, and we see the same thing as a result of hypergravity resistance training. So certainly hypergravity resistance training has the potential to be effective, but as I mentioned in the previous slide, it's probably devices more like the Tesh yo-yo device that will be the device of choice. Here's a really remarkable study that was published by Scott Trappy looking at the effects of exercise on a 6-month ISS mission. And I want to highlight the fact that the individuals on that mission did close to, on average, about 21,000 contractions as part of the resistance training program, with a range of going from about 10,000 on up to about 32,000 reps. And that was combined with treadmill exercise and also cycle exercise. So from my perspective, if you sit there and think about it, these individuals were exercising quite a bit. So what happened with the muscles? How protective was the exercise program? So here's data looking at muscle volume, and the reason I'm showing this is because this is kind of our marker, if you will, of what's happening with the cross-sectional area of individual muscle fibers. And we're interested in looking at this column here, which is the delta between the pre and the post flight data. And you can see on average that even with what I'm going to call a fairly robust exercise program, there was still significant atrophy, about 15%. So where does this fit into in terms of the data that was shown by Dr. Adams? And here's something that perhaps is a little disturbing. So that star is the data from Scott's study. And so this, I think, is a foreboding finding. Now, I should mention, some of you could come up and say, well, there have been a lot of ground-based studies that have used a variety of different devices and shown much better results. But what I'm going to say is that this study, right now, because it's 6 months of flight data that involved an exercise program, has got to be our gold standard. It's got to be what we're working from. And so I think we need to be very cautious as we proceed forward. Now, Scott mentioned in his publication that this program was a moderate intensity, and the reason for that is that the resistance training device was the IRAD instead of the ARAD device, which is up there now, and the IRAD wasn't able to provide the types of loading that they would like. And so he noted that greater loading was needed. I would add that from our perspective, and I think all of you would agree, is that the astronauts need to be tested on some type of a regular basis, and that based upon that testing data, then their training programs need to be adjusted accordingly. And as Dr. Baldwin has said to me repeatedly, when you sit there and think about this Mars mission, it's a whole new ballgame, because we're talking about flight studies that are 8 times longer in duration. So really quite a major challenge as we move forward and begin to think about developing the most effective countermeasures. Here's a list of the different devices: treadmill, cycle ergometers, ARAD device, lower body negative pressure, human-powered short arm centrifuges, and yo-yo device. And as I said, the yo-yo device clicks off all the major boxes here, and plus, when you sit there and take a look at the key engineering considerations in terms of space and mass and whether power is required or not, it seems to check off most of the boxes there. So from my perspective, I have to be unbiased in this regard, and it's devices like that, inertial devices, that I think are going to be the way to go. Here's a smart unit. I just want to come back and highlight this. And this is actually some data. If you look carefully at Dr. Hood's papers, although he talked about a reticulum, which undoubtedly exists, there's something that's very fascinating about what I'm calling the smart unit. And David actually had a slide, an EM slide, that showed the same thing. Here's the Z line. Here's our thick and thin filaments out over here. Here's the mitochondria. They always seem to be located in this stereotypic area located next to the Z line. Hard to see it here, but here's the terminal cisternae of the sarcoplasmic reticulum and the lateral sacs out here. And there's always this relationship between the mitochondria and the SR. And as I noted earlier, the SR is linked to the sarcomere at the M line through critical proteins like obscurin and ankyrin 1.5. So to me, as we move forward, something that I'm excited about is trying to figure out if in fact there are protein degradation pathways that simultaneously uncouple the connection between the mitochondria and the sarcoplasmic reticulum and the SR and the sarcomere, and then that somehow leads to a set of events that lead to the loss of all 3 of these units at the same time. Now, for those of you who grew up in the era that I did, back in the 50s and the 60s, you'll remember the early days of the space program, and it was exciting. We were doing something brand new every launch, with the goal ultimately of making it to the moon. And we had a very charismatic president at the time who really was focused on this. And so here's the first set of astronauts, and you can see John Glenn down here, Wally Schirra, Scott Carpenter, Gus Grissom, Gordon Cooper, Alan Shepard, and Zeke Slayton. And then the group in the back was the second slate of astronauts, who by themselves were not chopped liver. That include Lovell and Neil Armstrong, so really famous individuals. And of course, the group here in the front, the 7, was known as famously as the Mercury 7 and was highlighted in a very famous book called The Right Stuff, published by Tom Wolfe, and then ultimately made into a movie. And the cool thing for us is that if you sit there and think about it, the ACSM has actually had its own right stuff, being guided by Sir Dr. C.M.Tipton and his right stuff. And I don't mean to exclude any of TIP's trainees. I'm just highlighting the folks that have made the biggest impact in my career as I've attended the American College of Sports Medicine meeting over the years. And so I would like to finish by sitting there and saying that Dr. Tipton recognized very early that physical activity imposes unique stresses on a broad spectrum of cell types, tissues, and organ systems. And in so doing, Dr. Tipton recognized that physical activity plays a key role in shaping fundamental biological processes necessary for maintaining health and preventing disease. From Dr. Tipton's intellect, energy, foresight, and probably just as important, his mentorship, came what I will call the Iowa 7. And you notice that we replaced the NASA meatball with the Iowa Hawkeye meatball. So through the efforts of the Iowa 7 have come succeeding generations, of which I've been the benefit of through my interactions with all of you, but most especially Ken Baldwin. And when you consider Dr. Tipton's illustrious career, his passion for the American College of Sports Medicine, and the legacy he's left, it's clear that he has the right stuff. And those of us who've been touched by him have been truly fortunate. Thank you.
Well, I want to add my thanks to the Terjung and Baldwin for organizing this and including me. And it is an honor to be here trying to honor Dr. Tipton. Dr. Tipton was on my thesis committee, and during that time fueled my fires for exercise science. And then he served as the co-mentor for my postdoctoral training and has been a trusted advisor throughout my career. Whoops. There we go. I've been blessed to have a number of excellent graduate students and postdoctoral fellows, and today I have the pleasure of introducing James Rush to you. And he has also continued training great-grandchildren. Dr. Rush got his bachelor's degree and master's degree from the University of Guelph, and then went to the SUNY, State University of New York in Syracuse, and got a PhD with distinction, working with Dr. Terjung. So there's some kind of strange biological offspring here that he then came to the University of Iowa to do a postdoctoral fellowship with us for 2 years. He then went to Waterloo and worked through the academic ranks through professor and then department chair and a couple of associate deans. And he is currently a professor and dean of the Allied Health Program. When I first met Dr. Rush, he looked a lot like the figure on the left. I was a judge at ACSM of a poster he was presenting. And the excitement and pleasure he had in science was obvious to me then, and you can see it. And during the academic ranks there in the middle, he didn't seem to have quite as much joy, but as he moved to the dark side of administration, Dean Rush had that excitement. And it was fun this morning as I met him and saw his face. He's still got that excitement. He's a good man. And come tell us about your science.
Well, thanks very much. I'll just point out one little modification, and that is that although this is a fairly recent picture, it was taken about 2 days after I became a dean, and you can see that the degradation of the last 3 years is accelerated compared to the previous 20 that Dr. Laughlin referred to. So I guess there's nothing we can do about that. I would say just very simply that it's an extreme honor to be part of the symposium today and to be asked by the organizers to present. You've seen the lists of the many illustrious people that have come from this lineage, and there are many much more worthy than I to be giving this kind of perspective talk. So I do really feel like it's an honor to have been asked. I only met Dr. Tipton once. It was probably maybe even at that same conference you're talking about, because I think he was hosting a lunch for award winners. It was great. Of course, I've learned a lot indirectly from my mentors, Dr. Terjung and Laughlin. But this is a still shot that's taken from a series of videos that the American Physiological Society did called the Living History series. And if you have a chance, and if you haven't already, and you don't know Dr. Tipton personally, this is a great hour or so video to bring to life a really complex story in a way that's very revealing about some of the key principles that have underlaid the tremendous impact of Dr. Tipton's career. And a few of those that came out immediately from me that you might not catch from reading one of his review perspectives are his commitment to being a generalist even when the system was changing to really not favor a generalist opinion. And I think that was really part of how he brought innovation and collaboration as key elements of the way that he operated. Another one that came out of just the interview was his humbleness and his humility and positioning the accomplishments of others before himself. Another was his mastery of experimental design. He was a real master of the principle of taking something away from the system, surgically or mechanically or chemically, and measuring in a very metered way what the effect was on the process he was studying, and then replacing it and studying the adaptation in the opposite direction, and doing so really systematically to really understand the system and how it was functioning. And the other neat thing was his focus ultimately on practical applications in real-life situations of what he was working on in the lab. So I took those things from my research on Dr. Tipton and various conversations I've had over the years, and I tried to apply those principles in just selecting the bits of information I'd share to put together something around this idea of exercise is essential for optimal cardiovascular health. So of course we have a really good starting point. We have really well developed concepts and guidelines from some fairly reputable agencies that tell us what the ideal concept of cardiovascular health is. So this is the American Heart Association concept based on 4 ideal health factors and 3 ideal health behaviors, one of which is exercise, and has prescription types of guidelines for ideal cardiovascular health based on a solid framework of evidence-based research in the moderate to vigorous physical activity realm. And in turn, the application of that to prescription in the guiding document there is ACSM's own position stand on exercise-based recommendations across the various parameters that define what exercise is. I mention these only to establish the solid framework and decades of research that have really do support the concept that exercise is essential for optimal cardiovascular health, and to use it as a launching pad to some of the new and innovative things and the challenges to the concept of that these documents espouse. One natural point in the evolution, and I won't go into this in detail, but is that there's now a solid clinical case for fitness as a clinical vital sign. And if you're interested in that particular topic, perhaps you'd want to attend the symposium this afternoon from 1 to 3 that deals with this issue on should cardiorespiratory fitness be a vital health measure. The reason I'm showing this now is to look at the all-cause mortality curve between various levels of cardiorespiratory fitness, either from that document I was just discussing or from a recent review from a slightly different perspective of Joyner and Green, looking at the influence of exercise on cardiovascular risk factors. The main point I want to make from this epidemiological data is that in addition to what we know about exercise as a mediator of known risk factors, there's about a 40 or 50% effect of exercise that is not associated with the mitigation of other known risk factors. So you'd either call that some kind of direct effect of exercise or an effect of exercise on a risk factor we've not yet well defined. And so Joyner and Green call that the risk factor gap effect of exercise. And I mention it now because I'll come back to it later when I talk about mechanisms a little bit. But if we move to the actual determinants of VO2 max or cardiorespiratory fitness, we see this classic curve that we all use in our teaching, plotting oxygen consumption versus workload. And of course, there's a linear relationship, and where you are as an individual on this curve is defined as your VO2 max. You reach a workload you can no longer tolerate. With aerobic support, you can't consume any more oxygen because the variables that are limiting oxygen consumption have been maximized. We know that where you are on this curve as an individual is about half dependent on the gifts your parents gave you by mixing their genetic material together to create you, and then about half based on what your behaviors and your activity level is. And pioneers such as Claude Bouchard, the Heritage Study, and many others around that time were really instrumental in developing that concept. The point I want to talk about today is in the middle part of this curve, there's a tremendous capacity for plasticity for you to move up and down this curve of oxygen consumption capacity, or which is associated with cardiovascular health because of the activities you choose to participate in. And of course, to also note that what you see here in this very simple curve has a very complex underpinning that depends on the components of the Fick equation that is expressed here, and the many, many elements of the respiratory, central circulatory, peripheral circulatory, and muscle metabolic systems, the dozens and dozens of factors and thousands of proteins in some cases that are actually determining either the extraction or the central cardiovascular variables that are coming to play. So I go back to try to find a landmark study to try to build some case for. It's very apparent that this classic study, known now as the Dallas bed rest study, is a great example of one of the principles I was talking about later, and that's that taking something away and measuring responses and putting something back and measuring the responses in the opposite direction, or following somebody up and down that VO2 versus workload graph that we just looked at before. So this paper was, of course, published in 1968. And it asked two general questions. Of course, it took 78 pages to address those two questions. So this was an era where a 78-page paper in circulation was acceptable. And we're really lucky that it was. 35 figures and 22 pages of raw data in the back. Actually, an extremely valuable gift to everybody that's ever read after 1968. And I'll tell you one story about how that comes back just a couple of years ago. Two basic questions. What happens if you subject 5 fairly healthy young males, 20 years old, to 20 days of bed rest? And the answers are pretty clear. About a 25% decrease in VO2 max that was pretty much entirely the result of a decrease in maximal cardiac output, and that was a result of a decrease in maximal stroke volume. So that was the basic observation, not much of a change in extraction in going in that direction. Then what happens if after that bed rest you subject the same subjects to 8 weeks of dynamic cycling exercise, about 80% of heart rate max? And the answer to that was fairly clear as well. You recover all that you lost in a little bit more of the VO2 max, which occurred because of increases in the cardiac output max, and although it's not as easily appreciated from this graph, also significant increases in the AVO2 difference. At the time, those AVO2 difference increases were interpreted strictly to mean peripheral skeletal muscle adaptations, but it actually was Dr. Wagner did a reanalysis using that 22-page raw data supplement just a couple of years ago to go back and honor this significant contribution. And obviously with newer techniques and appreciation of the concept of what's going on, the relationship between blood flow diffusion and RBC transit time was able to make a diffusive unloading of oxygen argument. So it came back to pay dividends again. It also paid dividends with a 30-year follow-up study which addressed issues of aging and exercise training in the same individuals that participated in the original 1968 study. So 2 follow-up papers came out. They totaled only 17 pages in circulation, so times had changed in the 3 decades from the original paper, but they were split into 2 papers, one that addressed the aging issue and one that addressed the exercise training issue. So on the aging issue, the question is what had happened over that 3 decades of time. So depending on how you normalize the data or don't normalize the data for body morphology, there was about an 11 to 14% decrease in the VO2 max that occurred over the 3 decades as a result of aging in these fairly healthy individuals. That was driven primarily by a decrease in the AVO2 difference because there were increases in stroke volume that had occurred with aging that compensated for the decreases in heart rate max to make cardiac output max about the same as they were 30 years earlier in those same individuals. What's really impressive is this 11 to 14% drop in VO2 max that occurred with 3 decades of aging was actually only about half the size of the decrement that happened with 20 days of bed rest in the 20-year-olds originally in the study, showing the profound effect of the inactivity on cardiorespiratory fitness and health. The second component of that study was to do an exercise training study in the older individuals, the 3-decade-aged older individuals, and that was using about a 6-month intervention period with the goal of trying to achieve 75% of heart rate max in the training sessions and adjusting accordingly over the time. So what had happened, what that was able to do, now if we're looking at the far right bar compared to the second to the far right bar, was to recover the loss in VO2 max, 14 to 16% increase in VO2 max, so that the levels of VO2 max were about the same as the same folks when they were 20 years old, 30 years earlier. And that was driven by an increase in AVO2 difference with, again, changes in stroke volume max offsetting a heart rate max and cardiac output being relatively the same. So as all of us get older, I think this is one kind of really brilliant illustration of the concept that exercise is essential in optimizing cardiovascular health, in the sense that at a very simple level, this reversed the cardiorespiratory effects of 3 decades of aging. So I think collectively these studies are a fantastic example by manipulating in both directions up and down that VO2 max curve and studying what the physiological mechanisms underlying it were of this principle that activity levels and exercise are essential factors in controlling optimal cardiovascular health. Now the problem is that we have a societal problem with physical activity, and it's possibly illustrated well in this figure, that comes from the American Heart Association report, Sedentary Behavior and Cardiovascular Morbidity and Mortality, and is really based, this figure is based on the National Health and Nutrition Examination Survey. This little green slice in here is the kind of physical activity that's typically prescribed for cardiovascular health. Very, very small component of a whole day's worth of minutes and hours that we live every day. This is what we know the most substantial mechanistic-based evidence for prescribing exercise as a health behavior. But what is emerging is that the contribution to cardiovascular risk of the sedentary component of our daily lives is an extremely volatile risk factor, and the degree to which they're independent or interact is, there are some data, but is a topic that should be of really intense interest, because the epidemiological reports, as Dr. Booth points out, suggests that the regular defined bouts of exercise, the MVPA type of exercise, that may protect against early mortality may be vulnerable to counteracting effects of the increasing amount of sedentary behavior in our lives. And it really points to needing focused studies on the mechanisms of what the physiological adaptations to sedentary behavior are, so we know what we need to be doing in terms of avoiding sedentary versus achieving a certain level of physical activity in the mix between these types of behaviors during the day. Now, that's easy to say, and it's harder to do, and it's harder to do because of adoption of behavioral changes. And of course, one of the huge challenges is that people have with meeting current guidelines is the time to participate in such exercise. So again, an analysis by Dr. Booth points to the need for innovative solutions that draw on a strong scientific foundation to drive innovative solutions, both in exercise prescription and technologies that may assist. So one idea that comes out of his analysis is related to the known benefits from high-intensity interval training in the early years of that work on many metabolic-related parameters, and whether or not there's a general application of that to many of the other cardiovascular and other metabolic systems that need to be perturbed to maintain optimal health. We have a thought leader in this area who's actually present at the ACSM conference. So this is a figure from Dr. McInnes and Gabala's recent review on high-intensity interval training. And I do want to point out the major contributions of Professor Gabala, who resides just in proximity to where I am in Ontario. He's at McMaster University. He's shown international leadership in providing and evaluating the evidence in this area. And this article is a brilliant synthesis of the current state of the field. It's very recent. Now, Gabala is here giving a president's lecture on Saturday, June 3rd, at 11:15, just down the hall here. That is entitled Interval Training for Health, Hype, Help, and Hope. And I think it would be your distinct educational pleasure to attend that if you happen to be here still on Saturday. Gabala points out, and this is where there's the deficit in linking the early, very promising results in high-intensity interval training to metabolic outcomes. Gabala points out that the same type of evidence doesn't exist in excess in terms of central cardiovascular adaptation. So the jury's still out a little bit on the role of high-intensity exercise training on the cardiovascular adaptations that could support long-term cardiovascular health, but I really hope that this is an area that is stimulated by the perspective of people like Gabala and like Dr. Booth. And in fact, there's early evidence that that's happening. A paper that just came out in 2017 is trying to build a case for high-intensity interval training, increasing central cardiovascular adaptations that support VO2 max, and that's building on a background of high-intensity interval training with various other types of ongoing exercise afterwards. I won't go into the details, the citations here, but it is showing a consistent positive effect on VO2 max and stroke volume and cardiac output adaptations. The questions are, what are the mechanisms that are driving that? What's the sustainability of that effect over time? And does this address the question of the time management part of the problem with achieving desirable levels of exercise? Because if the supporting high-intensity exercise has to be long enough or voluminous enough, we're going to be in the same problem we are with trying to adhere to MVPA types of exercise prescriptions. There's lots to learn, and there's lots of good and positive things coming from this innovative approach. I'd be remiss if I didn't talk about the endothelium a little bit, because probably Dr. Laughlin would be very disappointed in me if I didn't. And this will come back to the point I was making earlier about Joyner and Green's perspective on the risk factor gap. So so 4 decades ago, the endothelium was considered to be just kind of in the way, maybe kind of protecting the blood vessels. But some fairly significant discoveries in the 80s spurred decades of investigation into the physiology and the molecular biology of the endothelial cell. And it became very clear that the endothelial cell was an extremely sensitive transducer of many types of signals, including mechanical signals. So I want to talk about that particular model, the endothelium as a mechanosensor, as a possible mediator of the effects of exercise in this risk factor gap. And that'll build on the decades that of research that have shown that, of course, this endothelium is now appreciated as a very important autocrine, paracrine, and endocrine regulator of cardiovascular health. And And furthermore, in more recent years, has been appreciated as a transducer of the chronic adaptations of the circulatory system to the stresses of exercise and other stimuli that provoke mechanical perturbations in the vasculature. I want to, coming back to the risk factor gap analysis, so Joyner and Green put forward two sorts of hypotheses. One relates to the autonomic nervous system adaptations, the other identifying endothelial cells as prime candidates to explain that risk factor gap in the effect of exercise and cardiovascular health, independent of the effects on other known risk factors. And I want to use just a couple of really neat recent studies that use some clever interventions to show this mechanical coupling and really support the idea of exercise as that endothelial stimulus. So the first one, and this is primarily from Padilla's lab in Missouri. I'll go through this really quickly because I think I'm getting to be over time. I guess that's what these lights mean. What they did at first, and this was very clever because they took regularly active people that were meeting physical activity guidelines and forced them to reduce their activity. Not to zero, so not a bed rest, not an immobilization study, but just reduced down to a sedentary level. And it was very clever because it was I think, one of the first studies to do that and created sort of a real world situation of understanding the transition from physical activity to sedentary behavior. It only took a few days before the popliteal artery flow-mediated dilation, which is endothelium dependent, showed severe impairment with this reduction in habitual physical activity over that time. The same was not true in the brachial artery, which was not an involved limb in the change in activity protocol that had occurred. So it was a specific local adaptation, likely to the mechanical environment that was created by the lack of activity. Similarly, in a follow-up study that was on sitting, sitting is associated with a profound drop in endothelial-dependent flow-mediated dilation in the popliteal artery, the lower leg artery. And that is thought to be dependent on the reduction in shear stress in the black bars here over time that occurs during a prolonged sitting. Now they did something very clever. Instead of exercising the leg, they wanted to just look if shear stress and the mechanical factor were causative. So they heated the one leg and didn't heat the other leg. And the heated leg, of course, resulted in increased blood flow through the popliteal artery to the peripheral tissues for thermal regulation. That increased the shear stress throughout the sitting protocol while the subjects were still sitting, and it totally recovered the flow-mediated dilation in the popliteal artery, suggesting that it was the shear stress deficit that caused the decrease in flow-mediated, the endothelial function. What better way to increase shear stress in a limb artery than to exercise with whatever limb is present? And this finally is my last slide, and just shows that the exercise snacking hypothesis that Booth has put forward, this idea that a little bit of exercise before some sedentary behavior can be beneficial in some context for metabolic adaptations, is also true vascular-wise. So pre-exercise subjects maintained higher levels of shear stress during a sitting protocol and protected their endothelial function, whereas those that just sat didn't. And the same was also true for standing. So I mention that only in the sense that these are real-world situations that could be occur occupationally or in the home, where these interventions that have to do with activity and mechanical stresses on the endothelial cells show profound effects on the cardiovascular health, and that those things all occur naturally during activity and exercise. So we just looked at some mechanisms, but bringing it back to the exercise scenario, I think that we can conclude from historic and from emerging and innovative perspectives on this issue that yes, exercise is essential for optimal cardiovascular health. It's rooted in the guidelines in the MVPA world of prolonged exercise, where we have a solid database. The evolving field has its challenges. Some of those have to do with lifestyle adherence, tolerance, changes in increasing sedentary behavior, driving innovative approaches either in exercise prescription or in technologies to address this. But it makes the case, as Dr. Tipton would say, I think, that even more important to understand the physiological mechanisms of the activity, the sedentary, and the exercise effects on the cardiovascular system to provide better guidance for the practical outcomes that could come from this. So I apologize for going a little bit over, and I thank you for your indulgence, and again, very happy to be part of this symposium. Thank you.
I too would like to thank Ken and Ron for organizing this symposia. I would like to point out that even though in the Lineage I've been listed down the line in PhD students, I was actually Dr. Tipton's first student in his exercise physiology program at Iowa. His concept was to take people who had a background in exercise, namely physical education, and to make them into scientists. And I can tell you, he was a strict driver of what it took to become a scientist. And I can assure you, I was totally unprepared for that program. But somehow I survived and went on. And as you can see, in the Lineage here, I've had a great career at UCLA, and was able to develop a PhD program in exercise physiology at UCLA, which we followed the Tipton concept of becoming scientists. And now I have the fortune of introducing one of my students, Chris Roberts, who is going on to produce some of his own students. Chris was in my program from '94 to '99, following graduation where he earned his MS and PhD degrees. He did 2 postdoctoral fellowships. He came back to UCLA for a while where he was director of the Exercise Physiology and Metabolic Disease Research Laboratory, carrying on the research that he started in my graduate program of understanding the value of diet and exercise for the prevention and treatment of chronic diseases. Chris presently has 86 peer-reviewed publications. He's had 4 additional papers in press or review. And now I'd like to present Dr. Tipton, or Dr. Tipton. I wish I could present Dr. Tipton, but I'll let Dr. Roberts do it.
Thank you, Dr. Barnard, for the introduction. And at the outset, I just want to say, you know, when people stand up here and give presentations, you know, we really don't know what the lineage of everybody is. And, you know, Dr. Barnard is like a second father to me. I met him in 1994. Not sure if he remembers this scenario, but I sure remember this scenario. And so we've had a relationship for 23 years. He was always extremely supportive, especially in the time 2008 till late in 2012 when I came back to UCLA. And for the first almost 5 years, UCLA didn't even provide me a laboratory. And to put that in perspective, probably there isn't one other researcher you can think of that didn't have a laboratory for 5 years and had more than $3 million in NIH direct cost funding. I also want to thank Dr. Tipton, who I was able to speak to on the phone last night, and he had some great things to say. Also to Ron and Ken, I'm actually very surprised that I was invited to be in this session because of the fact I haven't been in research now for about 3 years. But hopefully the data that I accumulated during my time at UCLA is worthy of our discussion here today. And so without further ado, what I'm going to do is I'm going to go over 3 main areas of research that I performed while I was at UCLA. The first is going to be the impact of diet on cardiovascular health. I know the title says exercise, but that was to get the session approved. But I'm really looking at diet, exercise, and cardiovascular and metabolic disease. Second, I'm going to look at the relationship of and the effects of short-term lifestyle intervention modifications on cardiovascular and metabolic health. And then third, which was really the focus of my laboratory when I came back to UCLA in 2008 for that 7-year period, was looking mainly at the relationship between resistance training as a cardiometabolic therapy. So let's start first with the initial studies, my first bullet point there, which were animal studies that I performed, and these were diet studies. And this was a model that Dr. Barnard had set up back in the 1980s, which basically looked at an analogous diet to a typical American high-fat refined sugar diet, and we actually called it HFS to stand for high fat and sucrose. And that was the diet here on this side. So it's about 40% fat and 40% sucrose. And then on the one on the right-hand side is essentially standard rat chow. Okay, so it's about 20% fat, 60% carbohydrate, and both diets have about 20% protein. And the first paper I published, actually, the reason that I ended up doing these studies was the first study that I ever did with Dr. Barnard was a paper that I published in AJP in 1997, which looked at the effects of acute exercise on the insulin and exercise-mediated glucose transport pathways. And we noted that nitric oxide, there was a paper in Nature in 1994 that suggested nitric oxide might be important in skeletal muscle, and a paper Tom Balon published also that was related to that. And so my first paper was on nitric oxide, and Dr. Barnard suggested that I pursue my PhD in the area of nitric oxide. I wanted to pursue my PhD in leptin, and I think he was right and I was wrong. And so this was basically my dissertation work, which is funny because the first time I ever presented this data was at a symposium in Baltimore in 2001, and that was soon after I got my PhD, and I'm thinking, well, why am I being invited to be in a symposium? Well, I was in a symposium with Harold Laughlin at that time. So this is how we get to know some of these people in the field. And going back to my introduction from Ken Baldwin, I met Ken Baldwin in my first ACSM meeting in 1997, which was in Denver 20 years ago. So it's kind of interesting how things come full circle. But this was basically my dissertation, the coup de grace of my presentation, of my doctoral period, and that was when I had animals on standard rat chow for 2 years. Yeah, they were on this diet for a long period of time. Their diet didn't change, but when they were on the fat and sugar diet, their blood pressure went up pretty much in a time-sensitive effect. The longer that they were on the diet, the worse the blood pressure got. And then when we took the animals after 2 years and we switched them back to a low-fat standard carbohydrate diet, their blood pressure completely reversed. Now, one of the things that we found was that it was very peculiar that this time course was so long. So then what we did is we took male animals and we did the same thing, and we found that the males got hypertension much quicker. So then we thought, well, maybe estrogen is playing a role. So we overectomized female animals, and again, when we had them on standard rat chow and they were overectomized, they had no effect. We gave estrogen, there's no effect, but when they were on the overectomy and the high-fat diet, they actually had an increase in blood pressure that mimicked what we saw in the males. And when we actually gave them back estrogen, then that was also reversed. We also showed that there was a lot, and these, I'm not going through all of the data, but there was a lot of data that I reported that suggested that oxidative stress was a key factor in this hypertension that we saw. So I started pursuing the role of nitric oxide, and all of you know how nitric oxide works in endothelial cells, being converted from L-arginine to produce NO, which diffuses to vascular smooth muscle, to activate soluble guanylate cyclase to convert GTP into cyclic GMP and induce vascular smooth muscle relaxation. And in fact, I was fortunate enough to have Lou Ignarro, who won the Nobel Prize in Medicine and Physiology in 1998, as a member of my committee during my PhD, although I think I met with him 3 times during the whole period of my graduate career. And of course, some of these are endothelium dependent, like bradykinin and ACH, and endothelium independent, sodium nitroprusside. So what we did then is we put L-name, which is a nitric oxide synthase inhibitor, in the drinking water of the animals. And we noticed that when we gave L-name to the low-fat animals so that they wouldn't be producing nitric oxide, their blood pressure skyrocketed. But the animals that had high blood pressure, when we gave them L-name, basically nothing happened, meaning they already had some type of defect in either the ability to produce or have a nitric oxide produce its physiological effects. So we subsequently noted that there was changes in reactive oxygen species, we had a decrease in kind of an ORAC measurement of antioxidant capacity, and there was protein carbonyl increase, which is a measure of protein oxidation. We also reported there was endothelial dysfunction in these animals. Then we basically came up with this scheme that what was happening was NO, if it was being produced, was being scavenged by free radicals and therefore unable to dilate the artery. And in addition, there was a decrease in nitric oxide synthase expression so that the animals actually weren't producing as much nitric oxide, ultimately leading to endothelial dysfunction and hypertension. In addition, we showed that this diet induced significant hyperlipidemia, so they had elevated LDL and elevated triglycerides. Shown here, this is the standard rat chow versus the HFS, and I'll come back to this group here, which is again the group that we switched when they were on the HFS diet for 2 years, and then we switched them back to the standard rat chow for 2 months. Another thing that we measured was we took a look at the change in body weight in these animals over 2 years, as well as the food intake, and you can see that the body weight increased, so the animals became obese, and there's this obviously this link between obesity and some of these conditions, and many people feel, I'm not one of them, that obesity is the cause of all this dysfunction, and I'll get to some of that more later in the presentation. What we found was that the HFS animals were eating a greater volume of food per day, but the amount of calories both groups were eating was the same. So what was happening is the HFS animals, when they were eating fat and sugar, that was being preferentially deposited as body fat, while the animals that were eating the low-fat complex carbohydrate, they were oxidizing all of that carbohydrate that they were consuming. So here again, this is the LFCC is always green, HFS always red. And we can see that they had decreases in glucose transport, increases in insulin, and of course the increase in blood pressure that I just showed, and then here's the body weight data. And then here is what happens when you switch the animals back to standard rat chow after 2 months. You get complete normalization of the effects that you see. And that happens for both glucose transport, insulin, and blood pressure. And there's some drop in body weight, but the animals that are switched for 2 months, it's not long enough to normalize their body weight, so they're still obese. Here's data that we did with a surgery fellow at UCLA who helped us hang aortic rings, Todd Reel, that we published in 1999, basically showing that, again, LFCC here in green, HFS here in red, significant endothelial dysfunction, not much difference in terms of endothelium independent, which is, again, the ability of the smooth muscle to react to nitric oxide. And then the orange, again, represents what happens when we switch the animals back to the low-fat complex carbohydrate diet. So they had normalization of endothelial function within 2 months. Then what I did was I started working with Dr. Barnard in some of his human studies. And this was using the Pritikin lifestyle intervention, which at the time was at Santa Monica. The Santa Monica Center closed, and now it's in Miami, Florida. But this is a diet that's basically plant-based, high in fruits and vegetables, whole grains, and then daily exercise. It incorporates both aerobic and strength training. And there's some incorporation more recently of interval training. But the intervention is very, in terms of exercise, is very individually tailored to the individual participant. And the first paper that we published on Pritikin data was in Circulation in 2002, where we noticed that there was an increase in oxidative stress by the 8-isoprostane marker, and that there was a decrease in, there was lowered levels of nitric oxide metabolites when they came to the center, but when they left it increased. So somehow, when they were going through this lifestyle intervention, they were producing more nitric oxide. And when we correlated the amount of nitric oxide excretion in their urine to the decrease in fasting insulin, we saw a pretty significant correlation, which at the time was a hot topic because there was this relationship between insulin sensitivity and nitric oxide. We went on to do cell culture studies looking at various fluorescent markers of either oxidative stress, that's here on the left. So this is a marker of reactive oxygen species. So what we would do is we would take the serum from the humans before and after the intervention and incubate it in cell culture, incubated with human aortic endothelial cells. And then we would measure from the supernatant or from various markers, or we would measure actually using the fluorescence what's happening in the cells. So after the intervention, something in the serum was inducing a decrease in the production of reactive oxygen species and an increase, this is a marker of nitric oxide production. We also did assays to show how many monocytes were being escorted to the area of injury. That's called monocyte chemotaxis, as well as what's called monocyte adhesion. And so this intervention was also improving these markers as well. The other thing that we began investigating was this concept that when you go on a very low-fat, high-fiber, plant-based diet, what happens is your HDL decreases. And that HDL drop has been the biomarker that we had the most problem publishing data from Pritikin. So most of you are familiar with the functions of HDL. It's very anti-inflammatory and antioxidative. It also is important for decreasing adhesion molecule expression and reverse cholesterol transport. So we collaborated with the atherosclerosis research unit at UCLA to start investigating this. And so what we found was when we took isolated HDL from individuals that went through the Pritikin program, when they came to the center, their HDL, quote unquote, good cholesterol wasn't good, it was bad. Okay, so there's this double jeopardy of HDL that basically says that when you have heart disease, your HDL may not be functioning properly. So their HDL actually was pro-inflammatory. It wasn't doing its job. And then when they left the center, they had this turnover of the deleterious HDL, and their HDL that remained and the new HDL was being produced was anti-inflammatory. We also looked at C-reactive protein and matrix metalloproteinase 9, which is a key marker of plaque stability. And this was actually shown not only in men, but also in women and children. The other thing we did is we did basically a summary of adding all the patients from multiple studies together to basically take a look at all the individuals that had come to Pritikin who had had high blood pressure. This was a study of about 1,100 individuals. They came to the center with a blood pressure of 134/79, and you may be thinking, well, that's really not that high. Well, these 600 of these 1,100 individuals are on oral agents to control their blood pressure. When they left the center, their blood pressure 3 weeks later, so they're on that lifestyle intervention for 3 weeks, their blood pressure, it drops, both systolic and diastolic, and more than half the individuals are no longer on any medications to treat their high blood pressure. Some of the ones that remain on medication have their dosages reduced, and of course that's beneficial because of all the negative side effects that are associated with these blood pressure medications. Then we looked at also in a summary of 6 studies of individuals that had come to Pritikin who had type 2 diabetes. This was at a time when the ADA criteria for type 2 diabetes was a blood sugar 140 or more. They had very poorly controlled blood sugar, 183. 284 of these were on oral agents to control their blood sugar, and 280 of them were on insulin. After 3 weeks, they had a decrease in blood sugar. 3 out of every 4 of them were no longer on an oral agent, meaning they had their diabetes reversed. It's not cured, because if they go back to their normal lifestyle, the blood sugar level is going to come right back up. And about half of them were no longer on insulin. And at the time, Dr. Barnard published a paper in 1994 saying that there was the need for early emphasis. So not only could we show the reversibility of diabetes with lifestyle modification within 3 weeks, but you had to institute the lifestyle modification as quickly as possible, because if you were at a latter stage when you were already on insulin, the chances to reverse the disease were going to be less. As I mentioned, we also did studies in women and children. One of the only pieces of data that I haven't published that I hope to publish soon is now the CMS has a new benefit classification, which is called intensive cardiac rehab. Classic cardiac rehab is 36 sessions of exercise. Intensive cardiac rehab is 36 sessions of exercise and 36 sessions of education. And the Pritikin ICR program incorporates both video as well as food demonstrations. So this is data from Oklahoma Heart Hospital, which basically shows that in these 1,280 patients that went through the center, went through the program, down here at the bottom is the number of patients that have this level of dysfunction. When we look at all 1,280 patients, the net effect is a drop in all of these markers in all of them, but the decrease is predicated on the level of dysfunction that they have when they come into the program. So in other words, those that came in with a cholesterol greater than 200 had a 14% drop in cholesterol. If their non-HDL cholesterol was greater than 130, a 17% drop. Their LDL was greater than 130, a 22% drop, so on and so forth. So these results that we're seeing were actually very similar to the results Dr. Barnard published in Archives of Internal Medicine in 1991 on more than 4,500 individuals that had gone through the center in 3 weeks. Now this is outside the center at hospitals in a 12-week intensive cardiac rehab program. And then we also looked at blood pressure. And in those individuals that had a blood pressure, 492 of these 1,280 people 492 of them had a systolic of greater than 140. Their blood pressure dropped 14% on this ICR program. And those that had a diastolic greater than 90, 213 of them had a drop of 21% in their diastolic pressure. So we hope to publish this soon. The other thing, again, with respect to the children is that this concept that obesity is so important. Well, one of the problems is that obesity is simply a marker of a poor lifestyle, and that does not necessarily mean that obesity is the cause of the problem. So what we decided to do is we took a look at children that had gone through Pritikin, and those that were obese are shown in green, and if obesity is the cause, then all the obese children are going to respond, and lean children or normal weight children aren't going to respond. Well, that didn't happen. Both groups responded the same to the program. And this was a 2-week program in children who came to the center during a summer. So they weren't doing a classical aerobic and resistance training program. They were doing more playing outside type of things. And then this was a battery of inflammatory markers that we measured, and again, lean and obese children respond the same. So it's the lifestyle that induces the metabolic dysfunction. So I summarized this part of the talk by basically saying that poor diet and physical inactivity induces metabolic risk regardless of whether you increase your body weight or not. You may have an increased metabolic risk with no increase in weight, or you may have an increase in weight, and therefore when your weight is elevated and you change your lifestyle, you may or may not have a decrease in body weight. And many people have shown this. So hopefully carry forward, future investigators will be able to study this more. And now I'm going to conclude with my studies on resistance training that I performed over the last few years at UCLA. And this really started with my interest in my personal interest in resistance training. Another individual I need to thank in the room is Frank Booth, who I wrote this article with him in Comprehensive Physiology in 2012. And this was a figure from that article that we took from MSSE, an article that's very rarely noted, but basically saying that individuals that strength train throughout their life, when they're 80, have the same level of power generation of a 50-year-old who's sedentary. So you can really extend that level of strength out 30 years, and that can really make a difference long term. So I became really fascinated with resistance training research, not for performance, but more for a cardiometabolic therapy. And so one of the studies that we did is we basically took 3 groups of individuals, people who had a body BMI less than 25 or a BMI greater than 27. These two were matched for BMI, and the 2 groups here were matched for training status. So they had to do a minimum of 4 days a week of resistance training. And of course, if the 2 groups matched for training have the same phenotypes, then training is more important. And if the 2 groups who have obesity are similar, then obesity is more important. And we were heavily criticized during the review of this paper because of the fact that these 2 groups here, both groups that were quote-unquote obese, overweight or obese, BMI 27 to 35, we were heavily criticized because these 2 groups did not have the same body fat level, but not one reviewer ever said anything about the fact that these 2 groups also had differences in body fat. And you want to make sure you remember that, because what we're going to find is that in virtually every marker that we measured, the 2 groups that are similar in terms of their phenotype are the 2 that are trained. And you may be wondering, well, why did you base it upon BMI? Why didn't you design the study based upon body fat percentage or visceral fat? Well, we did it based upon what the BMI criteria is. And if you've come to other symposium that I've been involved with, you know what my feeling is about the BMI. But the fact of the matter is that in virtually every measurement that we performed, whether it was a blood pressure measurement or a vascular measurement, the lean-trained and overweight trained individuals virtually had the same phenotypes for almost every variable that we measured. And so I'll skip through the rest of them here. You can look up the paper if you want. And so I had come up with this concept also that in this group, maybe we can also modify the redox properties of HDL, and that may be more important for fitness rather than obesity. And that was a study that I had hoped to get funded by AHA when I left UCLA. And basically we had data that we published in 2013, again showing the same thing, that training was more important than body weight status when you look at the function of your HDL. And we have unpublished data to show that one of the proteins in the HDL proteome, which is called haptoglobin, is mimicking what happens with our HDL redox activity. The greater the redox activity, the more oxidative stress there is in your HDL molecule. Now, what we did was we took those overweight, untrained individuals and we gave 12 weeks of resistance training with a personal trainer at UCLA's gym. And we noted that there really was no decrease in body weight. Again, this concept that in order for cardiometabolic benefits to occur, you have to lose weight. Therefore, if you gain weight, you're not going to see benefits. Well, resistance training is an intervention that typically, if anything, is going to cause a greater increase in weight. And it wasn't statistically significant between groups, but there was an increase in lean body mass that was offset by a decrease in fat mass. And we also measured trunk fat, and this was by the DEXA scanner that we had in our laboratory. We also did oral glucose tolerance test to show that in these overweight, untrained young men, that they had an improvement in their glucose tolerance. And if you read that paper, we also showed that there was, we used calculations to estimate improvements in insulin sensitivity. And so I came up with this concept in an article I wrote with Dr. Barnard in Comprehensive Physiology in 2013 that basically said that both aerobic training and resistance training can improve insulin sensitivity and decrease the risk of type 2 diabetes, but the mechanisms by which these two modalities improve insulin sensitivity may not be the same. What we did is we took biopsies from these individuals and we noticed increases in several proteins. I've listed 3 of them here: hexokinase, GLUT4, and AKT, all went up 30% with resistance training. The other thing that we did when I got some funding from Medtronic is we started using continuous glucose monitors. Again, this is data I haven't had an opportunity to publish, but basically we took individuals that were doing resistance training. All of these individuals were given the food for all of the 12 weeks of the intervention. So we can't say that this is due to their differences in diet. And this is like the first 5 or 6 subjects in the study. You can see we're already seeing a trend. So this is continuous glucose monitoring. So this is the 3-hour postprandial period after they eat a meal, where they're having a decrease in their postprandial glucose response with RT, but in the control group, if anything, there's starting to be a trend for an increase. And then when you look at the AUC, you see there's actually starts to become a significant effect. So to summarize, maybe you could play that video that I showed you before. So at this point now, what I'm doing is I'm working a lot at the West Los Angeles VA. This is actually a veteran. I train the veterans there. This individual turns 94 in September, and he is doing incline press with 30-pound dumbbells for sets of 8. So in closing, again, I want to thank all of the individuals that I thanked before, whether it's Dr. Barnard, Dr. Terjung, Dr. Baldwin, Dr. Booth, all of the individuals that I worked with. You obviously saw that my list of trainees is a lot less than everybody else who's here in the room, and that's another problem that we had at UCLA is I wasn't allowed to mentor any pre-docs, so I had to rely mainly on undergraduates and postdocs, but I was fortunate to get a significant amount of funding during my career, and thank you for your time. I appreciate it.
Well, we're basically at the end, but I want Dr. Booth to come up to give a framework from Dr. Tipton. And one of the things about this guy here is that he lives in the lab, and he was the one that would come in at 7 o'clock in the morning and leave at about 11, and he probably still does that. So, Frank, you're the one. Take care of Tipton.
So I want to thank Ron and Ken, who have been really true friends for decades. Before I get started, I want to acknowledge also, since acknowledgments is done, I wouldn't be standing here without Dr. Tipton. And acknowledge that he was more than an outstanding teacher. He was really a model mentor and a true human being. He taught me principles that I carried through my whole life. So, okay, I want to sum up, and I'm going to go slow. There are only 5 slides, but I want you to look carefully. These are the direct trainees. And I'm going slow because I want you to see where you or your friends might fit into the primary Tipton family. So I'm going to pause a second and just let you think. And some of the parts of the second slide I'm going to show are, again, in the left-hand side, and this has been shown before, and it's been very nicely put together by Dr. Terjung. These are the primary Tipton trainees. And what I'm going to do is I'm going to show you how the Tipton family grew. And one of the things I saw in Dr. Tipton, which was important, is how much he loved his kids, his family. And one of the things that's also important to acknowledge is that Dr. Tipton included his trainees, students, as kids in his family. I felt I was one of Tipton's children. And so as we go through here in the family tree, it is a true family tree. So here's the grandchildren, and it's hard to read them all, but I'm going to again pause. Maybe you can pick out people you knew to see for a second rather than fly through here. And so think about this. This is the first, these are his grandchildren. And some of them are highlighted who have spoken today. And now we have great-grandchildren on the far right. So, as I said before, Tipton really loved his graduate students. And Tipton would love all these people. And he's really proud. One of the things I want to say is Tipton would always use the word proud. I'm proud of you, even if you did something bad. He was proud of you, okay? So just take a look at this. This is amazing. The legacy and what's been passed down. And I think Dr. Terjung was the one who put these together, and I thank him if it was him. So the last three slides are Dr. Tipton's slides. I'm going to play Dr. Tipton because he couldn't be here. And we chatted on the phone, and we both agreed I would read the slides. I'm not going to give any comments, and I want you to think of this. The 10 points I'm going to make they're on Tipton's three slides are his words, not my words. I think it means more if you read it rather than I say it. But I'll catch up with you now. I'm deeply honored and humbled by the implementation of the symposium and commend the organizers, basically Ron and Ken and others, and the college for bringing the event to fruition. Furthermore, I would like to request that all individuals whose names were mentioned or listed on the previous slide, please stand and be recognized. So everybody that saw their name on the slide, please stand so everybody can get a feel for letting Dr. Tipton know what his family is here today. Thank you for standing. As mentioned previously, I, Dr. Tipton, was recruited to Iowa to develop a graduate program in exercise physiology, which was nicely described, I think, by Dr. Terjung in the beginning, that met the requirements of the College of Liberal Arts and the College of Medicine. And the comment was made earlier, when you came in that lab, you were like a medical student. He always liked to say, when I got in there, I wasn't supposed to digress, but when I showed up the first day, I had an overload of coursework, and he said, that's normal. You're going to take those courses. Okay, that was the way things ran. But that's the only hope to be the digression I make. So he listed 10 things that I know I experienced, and I want you to look through these. I'm not going to read this ones, Dr. Tipton. You know what they are. And let you just think for a second and think, which one of these do I know that he gave to me? So again, these are his own words, and maybe at some point someone can, these be saved and we can get them in some kind of a remembrance of these are the points that he really believed in, that he imposed upon the list of primary people that came through his lab. So the final slide. I like point 10. He was always demanding that I should question everybody. But I have a feel for all 10 of these, and I hope you saw them, and maybe some way we can put them into perpetuity some way, or let some of the organizers have a feeling for that. So Dr. Tipton, I hope I did a good job presenting your points. And I thank you for everything you've done for me. Thank you very much.
We're delighted that you all have been part of this symposium honoring Tip. And with that, I'll bring the session to an end. Thank you.